Let's start. Thank you so much, everyone, for coming today for today's Cell Size and Growth Seminar Series. So, today we have two exciting speakers, Neil and Anna, who are going to talk to us about how essentially cell size and geometry regulates chromatin, and then how cell size is controlled in cancer tissue or spheroids. All right, thank you. So we're gonna have Neil first. Take it away. Thank you.
Oh, for some reason I requested the sharing permit.
I
Okay.
think I was
Thank you.
sharing, and so yeah, it should be good.
Excellent. All right. Well, thank you everyone for joining today and also for inviting me to share our work today. Um, so today's work is mainly done by my current postdoc, Alex Bermudez, and also with collaboration with Dr. Jimmy Hu, who is also in this meeting. Um, and the work just came out a few months ago, so feel free to take a look at that the paper if you have any questions. So just want to first highlight all of the people who did the work, Alex, Zoe, and Dr. Jimmy Hu. So my lab is fascinated by geometry, and I'm a soft matter physicist by training. And currently, my lab is in the mechanical engineering. So today, we're going to talk to you about how we think about cell size or growth from the perspective of mechanical engineers and as a soft matter physicist. So one quote that really inspired me was from Darcy Thompson's on Growth and Form. The book said, cell and tissue, shell and bone, leaf and flower are so many portions of matter, and it is in obedience to the laws of physics. There are no exceptions to the rule that God always geometrizes. So that gave me a lot of encouragements, like say, a lot of times we think about physical science, it's just about equations and, things that are not living, right? But turned out in life sciences, it is still under the governance of physical principles. So when I look at cell shapes, when I look at the branch of trees or the pattern of leopard spots, we're really thinking about these forms are not just beautiful, but they're governed by physical rules. Especially one fascinating thing about epithelial cells is they look awfully similar to foam to in the eyes of soft matter physicists. And we actually found quite a few good parallel analogies between the two systems, right? The first thing is there's always a force balance and junctions. For example, the mechanical equilibrium among line tensions or surface tensions, um, it needs both to be met in both the physical system and the living system. And then that probably also answers why almost every vertex within the epithelial monolayer, you always see three junctions that meet at a single vertex. That's just a very simple topological rule. And then both of the systems, foam or epithelial systems, they have to fulfill energy minimization principle. Both epithelial tissues and foams, they minimize the total mechanical energy. Cells, they minimize interfacial energy from the adhesion and tension, while foams, they minimize surface energy. That's from the surface tension. However, today we're going to address an open question here is how biological and physical principles that jointly regulate cell size within monolayers remains poorly understood, right? So what we know is, yes, physical rules, they might provide some insights on how overall the cells they look or how they grow, but it doesn't give us the full picture. Biology is still one of the dominant regulation here. And then furthermore, how cell size itself regulates biological processes is also largely unknown. And then today we're going to focus on epithelial monolayers. So the first thing that we did for this project is my student at the time did the staining of this histone 3 lysine 27 trimethylation Ne3. And then this is in a confluent jammed epithelial monolayer. You don't see the cell boundaries just because we didn't stain for that, but it's a very fully packed, 100% confluent epithelial cells. And when we look at the histone mark level in this layer, we were so surprised. First, this is a single clone, meaning that they should be all genomically the same. But you see a starking heterogeneity distribution of the histomark level. None of the cells exhibit the same histomark level. Why is that? So the first thing we try to understand what could be the potential regulators. We did a attribution analysis. But first, just want to mention that the reason we care about histone marks is because histones and DNA packages, they regulate which gene or transcription factors to be expressed and which are going to be suppressed. And especially histone 3, K27ME3 is a repressive histone modification that contributes to the regulation of gene expression by compacting chromatin structure and making gene less accessible for transcription. So what we did is we first look at the cell geometry or the shape by quantifying every geometric features that we could think of, such as the area, shape index, parameter, solidity, right? And we also look at the shape of the nucleus. Um, and then we consolidate all these variables, morphological variables, into a nonlinear function. And then we train the function using the data that I just showed you, right? We also did a segmentation on the cell morphology. And then we found that we can actually capture or predict the histone level to some extent for every cell within the monolayer. So that means that even though the histomark level is so heterogeneous and it's seemingly random, but they're actually predictable. And then they're regulated by, or they can be predicted by just the cell morphology geometry. So they're not just random. And then, so what we did is, okay, if we are going to pick one morphological feature to kind of understand why it's a regulator, then, so what we did is we decided to say, let's start with the nucleus geometry. And in terms of the shape, let's start with the size of the nucleus or the area of the nucleus in 2D. So what we did was we look at the histomark level normalized to the DAPI signal, and then we found that it's inversely correlated with the nucleus area. The correlation is not really strong, right? It's about like it's moderate. It's about like zero point 0.3, negative zero point 0.3 Pearson correlation coefficient, and but the p-value is significant, and we are able to reproduce the results across different cell lines and also even in using in vivo tissues. Um, so what we know is this moderate correlation is relatively conserved across different systems. Okay, so this is kind of interesting. We also look at and the kind of the complementary mark. That's the K9AC. That becomes that shows the opposite trend, which is positively correlated with nucleus area. But today I'm going to focus on K27ME3 as an example because we have 20 minutes. So um, what we know is there are quite a few things that can regulate the histomark level of K27ME3. And then two major things are the histone modifiers, right? So, for example, we have EZH2 and UTX that can methylase or demethylase this histone mark. So what we did was we added the corresponding inhibitors. So like GSKJ1, DS3201, they can inhibit UTX and EZH1 and 2 corresponding respectively. And what we found is as soon as we introduce these inhibitors, the previously observed anti-correlation, that correlation that I just showed you in the previous slide, is gone, is abolished. So that suggests that the correlation or the regulation or the associations that I just showed you requires the involvement of these methylase or demethylase. So what we decided is say, okay, then let's try to continue chasing after one of them, right? So we decided to focus on UTX inhibitor. And then what we found was the UTX is then in turn regulated by the size of the nucleus. The reason we say that is because, first of all, we observe a correlation between the UTX level and the size of nucleus. But also we did a functional perturbation experiments. So in this experiment, what we did is we have a protein patterning experiment. We use stamp to create adhesion areas of different sizes so that we can confine or we can expand the nucleus. So what we found was when we confine the cells or when we confine the nucleus, we can lower the UTX level. And then in the same time, we can upregulate K27ME3 level, right? So this is a good example where if you actively perturb the size of the nucleus, you can alter the UTX level and the corresponding K27ME3 level. So this is one of the important evidence that shows nuclear size regulates the H3 K27ME3 level through regulating the UTX level. So the next question that we ask is, okay, so far we kind of identify the effectors, right? So we know there's a methylates, demethylates that are involved, and then we know that the histone mark level can be regulated by the nucleosides. But then let's try to go back even like more upstream is what controls the nucleus size variation. So that kind of goes back to the cell area because when you look at this, when you simply just plot nucleus area versus cell area, we see a pretty robust correlation between the two. And this is kind of conserved across different time points of the cell culture. And then we also observe this strong correlation across different cell lines and also in vivo tissue. And I want to say that this is a really common thing that has been discovered over and over, right? The strong correlation between nucleus area and cell area. But one of the interesting things that we're asking is, what could be the biological processes or the compartments that really facilitate this correlation? So you know that how nucleus is communicate or physically communicated or biologically communicated with the cell through quite a few things. The first thing is the nucleus cytoskeleton coupling, right? So what we did was we express a dominant negative cache protein, which is a protein that uh, within the link complex. And when we mutate this system, what we found is the link complex gets disrupted. And then in the same time, the Pearson correlation that you saw in the previous slide was then abolished, right? By a little bit, by like 10%, 20%. So, but this clearly shows that the nuclear cytoskeletal coupling is an important function that really helps facilitating the size coordination between nucleus and cell. So this can be also confirmed if we specifically perturb cytoskeleton. For example, if you add blebstatin to inhibit myosin 2, if you then what we found is the Pearson correlation can also be reduced. However, we found that nocatazole, which inhibits microtubule, doesn't really do much in epithelial monolayers. So we tried a relatively high concentration of nocatazole. We didn't really see any Pearson correlation change until we start damaging the cells. Okay, so this suggests that actin and also how actin is linked to nucleus is crucial for coordinating the cell size and nucleus size. But one of the things that we found really interesting is if you look into literature, yes, the cytoskeletal function in coordinating cell nucleus size is known, but there's also a new um, model that has been recently introduced is osmotic pressure. And this is something that got me really excited because as a physicist, we're very excited about this kind of molecular level physical forces. So in this picture, what was proposed was within the nucleus, you have this macromolecules such as DNA, right? They can exert outward pressure. But within the cytoplasm, you also have all different kinds of proteins that would exert inward osmotic pressure. And then what eventually determines the size of nucleus is the balance between these two pressure sources. So to test this relatively new concept, what we did is we did a selective permeabilization. We use a very, very low concentration of digitonin. We added this to the cell media, and then we can selectively permeabilize just the cell membrane. Okay, but we keep the nuclear membrane intact. So this is almost like doing dialysis. And after we finished the selective formalization, we then added hypertonic shock to the cell culture sample by just reducing the osmotic pressure in the cell culture media. And then what we found was upon this hypertonic shock, there are significant swelling of the nucleus. And then in the same time, the Pearson correlation coefficient was like really, really dramatically disrupted, right? But I mean, to some extent, this is not super surprising. This is because you know the size of the cell has already been determined and it needs to be fixed because everything is jammed. But in the same time, as you swell change the nuclear size, they all swell relatively uniformly. So the Pearson correlation coefficient is like almost entirely abolished. But this is really cool, right? Because how cells regulate nuclear pressure is through this exportin, which is a import channel that regulates what proteins go into the nucleus and then what stays in the cytoplasm. So if we inhibit the nuclear export, which is using the SLX drug, we found that this Pearson correlation that we showed you is also reduced, right? So this again suggests that the cells they actively regulate the osmotic pressure balance that I talk about, and then in turn maintains the size coordination. So the last question I would like to answer today is, okay, we know that nuclear size, cell size are coordinated, and then they can regulate histomark level. But truly, fundamentally, what gives rise to the cell size variation? Why do cell size even appear random in the first place? Because I showed you that there are different sizes of nucleus, there are different sizes of the cell, right? So the answer here is what really changes the cell size during cell crowding, let's say after you seed the cells and the cells they grow to confluency, is through size reduction. Because once the cells they reach confluency, they keep dividing. And then every time they generate a daughter cell, everybody needs to be squeezed more, right? So the cell density can triple even after the onset of confluency. So if you track a single, like a mother cell that divides into two daughters, what we found is that it always divides into two separate daughters with different sizes, right? One larger and one small. And then you can actually see that if the daughter was born larger, they remain large. This is kind of a typical growth law, right? Because larger daughter, they have a larger surface area. Of course, they can grow faster, right? And then so was the nucleus. It was coordinated. So basically, the reason you see some larger cells in the monolayer is just because they were born larger. And then because of the division process, it's not even. Okay, now, well, as I said, I'm a physicist, right? So we kind of like to ask questions after questions. And then sometimes you see this kind of annoying kid that keep asking why? And then we're one of those kids. And then the really last question we're asking is, okay, then why do we care about physical principles here? Because everything I said here seems to be governed by biology. So turns out the physical principle is here. If you plot the probability distribution or the histogram of area, and then you can find, you will see that all of these histograms, they show a very similar distribution. For example, if you look at the MDCK, the epithelial cell area as a function of area the histogram, they show this interesting log normal distribution. If you look at nucleus, they also show a very similar log normal distribution. You look at different, like say, mouse epithelium cell nucleus, Hecat cell line. They also show they collapse on the same master curve, right? And then all of these can be described by a log normal fit. The reason this is a log normal fit is um, this is what we call, and I like to use this um, 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 illustration to say is we have a golden board. And then every time you drop a marble through this golden board game, it will hit a nail. And then when the marble hits a nail, it needs to have decide whether 50% to go to right, 50% chance to go to left, right? So because they have to make this decision serially, eventually the distribution has to be a Gaussian distribution, has to be a normal distribution. This is just a collective behavior of a series of decision. Now, the reason we did not see log normal, I'm sorry, the reason we saw a log normal distribution, not a linear normal distribution, is just because every time the cell they make a decision, it's not about plus minus zero point 0.5. It's more about the fold change. It's either multiply by 1.2 or divide by 1.2, right? And that this is actually why because most of the noise within the biological system is multiplicative. So when the cells they divide, it's exposed to a series of multiplicative noise, and then that in turn gives you the size distribution that can be better described by log normal rather than just a typical Gaussian distribution. Okay. And then, by the way, we don't have much evidence to say this, right? So I would say this slide is relatively speculative. And then this is kind of just illustrate our current thinking of this question. Okay. A summary slide. What we showed in this work is the two histomark levels can be regulated by the size of the nucleus, and the size nucleus is coordinated by the cell, and then through the cell skeleton and osmotic pressure. And then eventually, why the cells are different is just because the cells they divide unevenly. Okay, with that, I would like to thank this wonderful, fascinating group again for doing this wonderful work. And then thank you very much and happy to take any questions.
All right. Thank you so much, Neil. As always, if people have questions, please type them out in the chat and I will read them out. So maybe I will start, Neil. So we actually work we so our I'm part of Jan's group, and we had also sort of really tried to think clearly about how cell size variation happens and is inherited in epithelia. And we had found that essentially the kind of the in kind of steady state tissue. So we were looking at like mammalian epidermis and how it turns over. We'd found that there the cell size variation is very much sort of driven and dominated by cell cycle variation. And that cell cycle variation is very tightly coupled to cell size variation. And I think Margaret Gardell's group also had found that essentially, yeah, that the in the kind of there is this kind of non-steady state reduction of cell size during crowding, and that during this crowding step, you get this kind of what's the word like uh, you sort of you seed all these heterogeneities through the history of the system, but then but eventually they found that if you watch the steady state part of the whole thing, the the cell size kind of evens out using the coupling to cell cycle. And I'm wondering if then kind of have you looked at the variation of the transcriptional mark, the histone mark, the epigenetic mark with cell size, and how is it if you look at the steady state and non-steady state parts, do they all follow the same line? Like, are they eventually is everything kind of collapsing onto the same sort of control variable, or is there, like the NC ratio, is it also changing with the crowding steps?
Well, this is a really good question, but also it's a lot of information. Um, what maybe I can try to focus on commenting on the histomark level variation. Well, so the first thing I want to point out is the correlation between histomark level and the cell size or nucleus size is relatively moderate, or I would say even weak in
Mhm.
our case, right? So I would definitely think it is very possible that the histomark level can oscillate throughout the cell cycle progression. So I think that would be something interesting to do. The only challenge that we're having is um, it's not that easy to live imaging the histomark level. There are not
Mhm.
too many tools available, but we would love to do that. Um, so so that's one of the challenges. But I think that that will be something really, really interesting to do. Um, in terms of are you also suggesting that the size of the nucleus or the size of the cell can also fluctuate or like oscillate um, um, uh, in the cells that are like actively undergoing cell cycle progression? Is that what you mean?
Yes, absolutely. Yeah. That the
Yeah,
even in the crowded state, if there is basically there's growth, if there's replacement and essentially turnover in space, that allows for um essentially growth, and that in that situation when the cells are hyperpolarized and really dense, the steady state turnover there the cell cycle is much more like very, very like tightly coupled to cell size. And that
absolutely.
yeah.
Yeah, so that's a very interesting question. In the particular cell line that we used, only about 5 to 7% of cells they are kind of like unsteady, like what you said, like they either undergo like some turnover, extrusion or newly generated. Um,
Yeah.
so that's kind of so we didn't really look into that
Yeah.
subpopulation carefully, but I think again that would be something interesting. And I we do recognize the paper from the Gardel lab. It's like a really beautiful work. So, but again, that will be something interesting to also look into.
Yeah. Um, I think Matthew had a question. Matthew, do you want to ask it or
Of course, yeah. So thanks again for a really nice talk. I wanted to ask you if you'd had a chance to look at anything that could be a proxy for global transcriptional changes in non-confluent cell culture experiments, as well as other model organisms. There's this clear correlation between cell and nuclear size and the global transcriptional output. Bigger cells transcribe more. And of course, that makes sense with a decrease in the intensity of a repressive epigenetic mark. So I was wondering if you thought about that, and whether maybe some other modifications you might have looked at could also, speak to something in that direction.
Yeah, well, this is a really, really good question. By the way, our collaborator Jimmy is also here, and Jimmy, feel free to chime in anytime. I think the two follow-up experiments we thought about is we would like to look at the overall chromatin accessibility. So for example, we thought about doing a spatial ATAC-seq, which is very expensive, but this is something we always want to do. There are also something we can look into is there are specific genes that are known to be associated with histone mark. Um, let's say K27, EMI3. So we could potentially do some like immune precipitation testing, or specifically to look at the expression of these either transcription factors or genes. So that will be something we can look into. Um, yeah, I don't know if that answered your questions, but and Jimmy, feel free to chime in about that part.
What I was going to say is, some of the experiments that have been done before are using approaches like EU labeling or immunofluorescence against the active forms of RNA polymerase or histone modifications that are associated with active promoters. And just it might be interesting to look at some of those global proxies for transcriptional output of a cell to
Mm,
see if they couple in the way that I was suggesting. But yeah, it's super interesting. Thank you.
absolutely. Thank you.
okay. We have a question from Raghav. And if you want to speak, or you can tell me to, I think I said that I would read them, but if you want to read them.
Yeah, okay.
Right.
So my question is the curve you showed, which shows that nucleus to cytoplasm ratio is constant. Is that only true for epithelial sheets, or is that true for every other human tissue?
Oh, I mean, this is a really good question we always get, right? So, um, well, I mean, even though I show you the that master curve across, like say 2 to 3 different cell lines, and also this mouse ear epithelium, but we also thought about like extending this observation to like 3-dimensional tissues. But when it gets to like 3-dimensional tissues, imaging becomes kind of challenging, and we're not talking about area anymore. We're talking about volume. So the quantifications imaging becomes exponentially more challenging. My so this is my personal speculation is I believe that NC ratio would probably still be conserved even in 3 dimension, but validating or testing that is requires some tool development. So that's my response to your question.
Okay, thank you.
Okay. We have I just wanted to note that I also we do have volume measurements and we have some that yes, the in
That's
volume
cool.
it's very
Yes,
much conserved. Yeah.
yes, I presented at the right community, so this is
Yeah.
great.
Yeah.
I can quickly read through other questions because I know we also need to get to Anna's talk, and I don't want to be the person taking too much time. So, do you want me to continue quickly answer those questions, or should we just move on?
Oh, I'm sorry. Let's move on.
No problem.
And people who have further questions, if you can just ask them in the Slack, that would be great. Okay. Thank you so much, Neil. Can we have Anna's screen?
Yeah.
All right.
So I will just share and start.
All right. Thank you. Take it away, Anna.
Yeah, great. Yeah, so first of all, also, Neil, many thanks for that interesting talk, and maybe there's time for discussion even later. But I also, of course, like to thank the organizers for inviting to present today. And this has been some team effort. And the main person that was driving this was my PhD student, Vaibhav Mahajan. He is now a postdoc in the lab. Can you all hear me? Yeah?
Yes.
Yeah, so before I come to my data, I also quickly want to present you my lab. So we are based in Dresden at the Biotechnology Center of the TU Dresden. And this is my group, and this is Vaibhav. And principally what we are interested in is basically 2 major things. So first of all, we like to understand how cells do respond to mechanical cues in 3D. And particularly, we are focusing on cancer. That's why we are the group of oncomechanics. And to answer these questions, we also employ a couple of different bioengineered models I will also touch on today. And a major interest is really also to see also how this level of adaptation also affects the mechanical properties of these cells. And here we basically like to understand also how single cell mechanical properties contribute to emergent tissue properties. And for that, we also use a couple of different techniques in the lab, starting from atomic force microscopy. We do some high throughput real-time deformability cytometry, what you can see here. And we also do more and more Brion microscopy. And I will also talk about 2 of these techniques today. And we also like to do these measurements, of course, on patient-derived tissues and cells, and try to understand whether we can also predict patient outcome, basically with these mechanical parameters. And yeah, so you might wonder why we are interested now in these mechanical aspects of cancer progression. And yeah, for this, I want to show you these H&E slides as a motivation, basically, that show prostate tissue, basically normal tissue. And then we see tissue that you see is quite distinct now for different tumor grades. Yeah? So these increasing tumor grades would be associated also with differences in patient outcome. And of course, you can probably appreciate that these development and progression of these tumors massively reshapes somehow. Yeah, so the whole microenvironment of these cells. And we also know that that there are a lot of mechanical alterations, and these go from single cells up to the tissue level. And of course, you see that this order of these epithelial structures is progressively lost, and we also have different interactions with matrix, other cells, and so on. And I really just, because I think this is known for many years, but I just quickly want to summarize that very quickly, what is known about these mechanical alterations, because of course, if you would talk to a clinician, they would also see that, because of course, a lot of solid tumors are felt by palpation. Yeah? So that's pretty obvious. There are mechanical alterations. And if we view at that from the tissue scale, we typically find that tumors are stiffer compared to healthy tissue. Interestingly, if you look at the single cells, however, we often find that cancer cells are less stiff compared to healthy cells, and if they are invasive, they are typically also less stiff. And there's another interesting aspect, which also, of course, has to be studied, or is better studied in 3D, I believe, that we also can have growth-induced compressive stress. And basically, the questions I want to get to today are, first, how do these tumor cells adapt their mechanical properties and also volumes in confining microenvironments? So this can be a stiff matrix, or it can be also in the context of a multicellular cluster. And how do these mechanical properties or volumes and mechanics, let's say, change once we acquire like an invasive phenotype? In order to address these questions, what we typically do in the lab, we use these bioengineered 3D tumor microenvironments, and that displays a bit how we make them. So we rely on biomaterials that let us also tune mechanical properties of that tumor microenvironment, but we can also make these hydrogels degradable and also equip them with specific adhesion sites. So this one here we do in collaboration with Carsten Werner's group. He's also based in Dresden. And so the way this gel is done, basically this hydrogel, is that we have a star pack and a maleimide functionalized heparin component, and we mix them. And by a mild click reaction, we now get a covalent hydrogel, basically, where we can embed our cells. And then we can basically adjust elastic modulus of these gels. They're predominantly elastic for now, and kind of adjust them to values that are kind of meaningful also for tissues. So this also shows, so we can also start with larger clusters, but in most cases, we really start with single cells that are embedded. So in black around, you would have the hydrogel, which you can see. And then we have here the F-actin cytoskeleton and the nucleus stained. And we see that over the time course of 2 weeks now, we get first to small clusters and then to larger size clusters. They're about 200 micrometers. And of course, we can also do some stainings now, like frozen sections. And what you see here is indeed we get a very dense cell mass. And around here, you see a bit the hydrogel. It's a bit of an artifact of the freezing. But what you can maybe appreciate if we compare that to a ductal carcinoma in situ tissue section, that there are certain similarities now. And what Weibach then did, he now changed the mechanical properties of these hydrogels and then viewed at these spheroids basically at 2 weeks. And what we typically find is that the growth of these tumor spheroid structure is massively reduced if we go now to a stiff hydrogel. So if I say now stiff, it's about 10 to 15 kilopascal, and a compliant gel would be 1 to 2 kilopascal. And at the same time, we also see that the spheroids are also getting more compact. So we also see that the nuclei are getting closer together. Yeah? So here we simply counted the nuclei and divided by the overall spheroid size. And we also see that if we now make these gels non-degradable, this is even more enhanced, this effect. So we even get to smaller spheres. So of course, we suppose that that had to do with compressive stress building up once these spheroids basically grow. And to show that, we teamed up with a colleague, Salvatore Gerardo. He made these beautiful polyacrylamide bead sensors. So these are polyacrylamide beads that we characterized mechanically. We put them together with single cells in the hydrogel, and then we get the spheroid growing. And then we see, especially in the stiff, you can appreciate that in proximity of the spheroid surface, we now get these bead deformations. And from these, we can basically calculate a radial stress that would induce such a bead deformation. And if we plot that over the spheroid diameter that we obtain at 2 weeks, we see indeed that there is an inverse relationship. So that we get the largest spheroid diameters when we have also the lowest radial stresses. So of course we thought, okay, there is no compressive stress, so would these cells now adapt also their own mechanical properties in this context? And so the first approach that we did is we basically isolated the sphere. So we can do that because we can degrade the hydrogel. So we got isolated spheres, and then we could probe those with an AFM tip, where we simply poke them with a little bead that is basically glued to that AFM cantilever. And then we can kind of read out an apparent Young's modulus. And what we see is that the spheres, but also if we do that same for the cells that we isolate from the spheroids, apparently change their mechanical properties once they are grown in a stiff hydrogel and compared to a compliant one. In any case, we were wondering also how that procedure of taking out these spheroids would affect the mechanical context. So for that, we decided to turn to Brion microscopy, which had been set up basically in the lab by Raimund Schlüssler. So he's now working at CellSense, a company that tries that actually commercializes this technique. And the way how this works is basically you scan your sample, and this is really all in situ now, and that is the great advantage. So you scan with a laser your sample, and then due to Brion scattering, you can pick up a frequency shift of the incident laser light, the Brion frequency shift, and thus we can now determine for each position and plot as a map. So that means we can map basically the elastic properties of a spheroid within the hydrogel without removing it. And this is because our Brion frequency shift is related to a longitudinal modulus. So it tells us something about compressibility of that sample. So Weibach employed this assay now on spheroids that had been grown in compliant and stiff hydrogels. And what you can see is that indeed we get increased Brion frequency shifts for spheres in the stiff, which would kind of corroborate the AFM results. He also, I mean, I will get a bit to the method later, but he also segmented nuclear volumes now in these day 14 spheroids. And just to highlight here that indeed we seem not to have nuclear volume changes of single cells. However, of course, it could be that we basically have a more compact structure because, as we had seen before, these cells come closer together, or it could also be that we have adaptations at the single cell level. So in order to get a bit more insights here, he did that experiment where he first of all looked at single cells in compliant and stiff hydrogels, then spontaneous aggregates, and then spheroids that had been grown in different contexts where we know that there are different levels of compressive stress. And indeed, you can see that the Brion frequency shift now scales with this level of confinement that we have. And interestingly, we also see changes now for the single cells, for instance, here in response to matrix stiffness. So that kind of let's suggest also together with the AFM results that in stiff matrices, we have mechanical adaptations now of these spheres. And at the one hand, we have a compaction of the spheroid structure, but we also have apparently cellular adaptations. So we also, Weibach also looked a bit more closely into these mechanical adaptations now of the single cell. So here you see plotted the Brion frequency shift of the cell over the gel, and you see that there is kind of a linear relationship now between the mechanical context of the gel and the cell. And also to let you know here that that was a bit puzzling for us also that, so in this case, he also segmented the cell volumes with LimeSeq. And what you see here is that indeed, it seems that there are no changes now between a compliant and a stiff hydrogel. So we get similar sized cell volumes at day 1. So that means that we see indeed also at the single cell level already a mechanical adaptation in stiff matrices, but this does not really affect the volumes. Nevertheless, what really puzzled us is now that we get, I mean, compaction is one thing we can understand in multicellular spheres, but in a way, these single cells should be the most compact structure we have. So we did not quite understand why we have this massive change now in the Brunner frequency shift if we go from single cells to clusters of cells. And that is something we then, that Weibach then looked more into. So he basically looked at these structures. He also included like an intermediate time point, day 4, and basically mapped them with Brion microscopy. And what you see is that indeed we have for compliant and stiff matrices both. I mean, there are bit differences as I have shown you before, but nevertheless, we get this massive change in the Brunner frequency shift now when we go from single cells to clusters. And we already thought at that time that this might have to do with nuclear volume changes. So this shows a bit better how he basically segmented with the help of Keshav and Byung these nuclei, and so using STARDIST. And at the beginning, I didn't want to believe it, but you see that indeed, when you come from single cells to small clusters and larger clusters, we get this massive decrease in the nuclear volumes. And this is true for the compliant matrix versus stiff matrix, and also in the stiff matrix. And we also see it if we now have free-floating aggregates. So what Weibach did here, he just put cells, 300 cells or 600 cells in a UVal. He let them compact for 2 or 3 days, and then he basically measured their nuclear volumes. So that was surprising to us because it meant that it had nothing to do with the mechanical confinement that we get these large nuclear volume decreases. So and I should also mention that we also don't seem to see what others had reported before. So we don't see any visual gradients now in these nuclear volumes if we go from the core to the periphery of a spheroid. So here you see the quantifications. So of course, we were quite puzzled because a 60% decrease in the nuclear volume is quite extreme. So of course, we thought about cell cycle, because we know that our nuclear volumes will fluctuate a lot along the cell cycle. And this is also visible here in this rather coarse time lapse that he did on a spinning disk microscope on repeated days. And indeed, to look a bit more into cell cycle, we decided then to turn to MCF7 cells that had carried this Fuji cell cycle marker, which allowed us now to read out by the fluorescent signal the cell cycle stage these cells are in. And to make it short, we see that indeed we get an accumulation of G1 cells in these later cultures. Yeah, so we have a bit of an abnormal accumulation of these G2 cells. And then with a small cluster, we get more G1 cells, and this is even going on. And he also quantified nuclear volumes. And you see, of course, as we would expect, quite some differences now in nuclear volume in G1 and G2. And we see that, of course, there is a huge effect now of this accumulation of smaller sized G1 cells. And together with the Honigman lab, we also looked into with light sheet, basically, more detailed now into these changes in cell cycle in a cluster, from which we could also get out basically the cell, G1 and G2 cell cycle time. So this was a little project we did then together with Sebastian Ahland. He's also based in Dresden, where he basically could also theoretically show that, of course, if we now feed in our cell cycle progression times from the light sheet, we can, of course, also replicate basically that a whole population would basically shift in average cell size if we have now a longer and longer G1 cell cycle, which seems to be the case. And in order to look at that more experimentally, so Weibach also did experiments where he applied the CDK1 inhibitor, which basically, so he applied that at day 4, and you see that we have more G2 cells now after drug application. And when he analyzes the nuclear volumes, we see that this can partly rescue also these nuclear volume decreases at day 4. So that was interesting because it kind of showed that indeed we shouldn't underestimate cell cycle in this. And if we have shifts in cell cycle, of course, this can largely affect our average volumes that we measure. However, we also realized, and maybe I should go back here, that we also didn't fully rescue our volumes to the day 1 volumes. So that also let us look into reversible events. So here what he did actually, he kind of removed these cells from the gels and from the clusters. And what you see here at a relatively short time scale, so this is about half an hour, 40 minutes, he sees that there's a fast recovery of some of these nuclear volume shrinkage, what we had seen. And of course, we don't know exactly mechanistically how this is going on. So we have started to do some more experiments, but so far, of course, what we suggest is that this has to do with swelling somehow once these cells come out of the cluster, which could be maybe due to osmotic regulation. So that would also be something that would be great to discuss. And we have some insights actually from ODT measurements, which we did together with Kyun Kim from the MPL in Erlangen. So what we could do here is we could measure refractive index in single cells in small clusters inside of the hydrogel. And what we see indeed, we have a higher refractive index on average for the clusters, which kind of is in line with a more crowded cytoplasm. So I think I'm a bit over time already, but we also looked at the reverse process where we also looked at a cluster where we had single cells shedding off. And indeed, what we see, and this is, of course, in line also with Minguo's work, we see that indeed cells that are kind of leaving a cluster apparently are more compressible. So we have a lower Brion frequency shift, but importantly also, we see that there is a kind of swelling happening here at this edge. So this is what we can see in vitro in our bioengineered model. But interestingly, we can also see that in patient tissues, where we of course can only look at nuclear size and not volumes in these H&E stains. But also here we have a tendency in the outer regions to have larger nuclear sizes. So I come with that to a summary. So we see that tumor spheroids get stiffer, basically, independence of microenvironment stiffness, and their cell scale mechanical adaptations, which we also still have to mechanistically understand a bit better, and also compaction apparently contributes. And interestingly, that response to stiff matrices seems to be independent of nuclear volume changes. However, when we track single cells, going from single cells to multicellular clusters, we see quite some drastic decreases in nuclear volumes, and these come along with mechanical alterations. And we see that contributor, which is major, is cell cycle shifts, but we also have reversible nuclear volume changes. And for invasive cells, we see also that they adapt their volumes and mechanical properties during invasion. So, of course, there's a lot of stuff we want to follow up on this. So first of all, to get more mechanistic insights. So we believe that, of course, osmotic regulation would be relevant. And also we should probably understand better how volume regulation actually happens in a cluster of cells compared to single cells. And of course, also, what are the effects on cell function, chromatin organization, and so on. So with that, I come to an end, and I want to thank a lot of people that were involved. And yeah, I thank you also for listening. And yeah.
Thank you so much, Anna. As always, put your questions in the chat. I will start with one, which is, I'm really interested in kind of the cell volume or nuclear volume dynamics as you know in these light sheet movies. Do you see essentially that like basically that there's is it like if there's like a size median sort of size thresholded or some sort of separatrix between the two cell cycles, and basically as the cells sort of compact more and more, there you get more G1 because you're sampling now more of the smaller G1 sizes. And is that something is that a model that's compatible with your data at all?
Yeah, so if I understand that correctly, so I didn't show these longitudinal changes now in the cell volume.
Yeah.
Unfortunately, for the larger clusters, we were a bit limited in the light sheet data because that's oblique light sheet. So we get so segmentation on these was a bit more difficult than on the static cultures. So yeah, so most of the data, the quantitative data I've shown you on the volumes actually, these came from cultures that were fixed, not fixed actually, they were viable. So we basically had a mCherry H2B expressed, and we included also like a membrane stain. And from these we could confidently do the 3D segmentations only on the nuclei, not so much on the cells, which is a bit of a limitation I forgot to mention because it's much more difficult to segment for the cell bodies compared to the nuclei. In the light sheet, unfortunately, we don't have very good dynamic data now on the volumes later on, but at the early traces, we can see indeed we can follow the volumes over the cell cycle, and there we see that I mean all cells seem to do. I mean, I didn't really show these single traces, but indeed we see that over G1 and G2, we see kind of a steady increase of the cell volume. And then, of course, we use the fluorescence data in order to know in which cycle stage we are. Yeah, and that is interesting. Also with the denser clusters, we still see that over the cell cycle also the nuclear volumes would increase. Yeah. So
Yeah.
it's not, I mean, you mentioned that Gadel paper, for instance, and that of course let us also think if we have at some stage a size reduction because maybe we don't have size increase between and then we have a division and that would be limiting now. But that seems not to be the case. So we
Yeah.
see that also later on the cells seem to increase all in volumes before they divide.
Yeah. Well, yeah, it's a
Yeah.
balance between, yeah.
Yeah.
Okay. We have a question from Gabriel.
Mhm.
I think you already answered my question. Very interesting talk. Thanks. It's hard to measure that, right? Yeah. The cell volume.
Yeah.
Yeah.
That is really tricky because
Yeah.
especially I mean, we can do that in the single cells. So that is relatively straightforward because
Yeah.
they have a quite round shape actually. But yeah, for the dense clusters, we really failed. So what we do is we can refer to an average, but this is of course also probably overestimating because we are supposed also, of course, we have also little gaps between the cells. So we see that if we do TM or if we do I mean, maybe you saw in some of these images, of course, there are also spaces between cells, and that is, of course, quite inaccurate. Yeah. But of
Can
course,
you dissociate
a rough
them?
estimate. Yeah. So we see also that, yeah.
Can
Sorry.
you dissociate them? Like,
The cells?
or
Yeah.
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 And
So
then,
can
like, could you
dissociate
measure it
them.
then better?
Yeah, I mean, that's what we kind of did.
Yeah. Okay.
But
Sorry.
yeah, maybe that was a bit fast. Sorry. So these were all dissociated.
Okay.
Actually, single cells, and this works.
Okay.
But of course,
Yeah.
we see that this recovery, of course, it would be much better to look at that in situ. Yeah.
Okay.
Yeah.
Thanks a lot. Very interesting.
Thank you. Ivan, would you like to?
Yeah, very interesting talk. Thank you. I just want to understand better your floating free floaters experiment because I think it's an important one comparing to a simple explanation that basically increased pressure from
Yeah.
the matrix slows everything down and makes it smaller. So, but I didn't quite understand what was different in that. If you can briefly explain a little bit more.
Yeah, no, it's a very good question. So, so the idea behind that was because, of course, initially we thought exactly we have compression, and that somehow lets them become smaller. Yeah, but I mean, of course, if we think about because we have measured these pressures and they're in the kilopascal range, so also theoretically it's really not enough, I guess, to drive these major volume changes in the cell. But if we can also show that experimentally, because here we have a stiff matrix, and we can even enhance that. So Weibach even did experiments where he used a non-degradable hydrogel, where we really know that the pressures are the highest. I mean, again, in the kilopascal range. And we still see that we get to very similar nuclear volumes at day 14. And the free floating is actually something that we also did in order to exclude, because before we always said, is it now with spheroid formation or is it with culture time? Or maybe who knows what that effect is. And these are created much faster, not as fast as we want. So we work now on also free aggregates that are compact.
So in these
one day.
free floaters, you get like individual cells and then you put them in that matrix, or what is happening?
We basically take single cell suspension, we put them in a U-well, which is kind of a low attachment bottom, and that kind of brings these cells together. But then it still takes, dependent a bit on the conditions, whether you include more matrix from the outside or how long you wait. The cells would find each other, bind to each other, and contract somehow, and this would form like a round spheroid structure. So this basically is shown up here. And we also thought, okay, maybe we should also see whether that is related to the number of cells we got. And this is an important point, because I think the most changes we see are really in here at the small clusters, but we seem to get to a threshold. So maybe that comes back also to Shikong's question. So we seem to reach like a certain threshold, which we seem not to exceed. So whatever we do, if we also have more, I think we even had one with 800 cells, we get to very similar nuclear volumes with that approach. But yeah, just to bring it back, I think it's not to do with forces from the outside, but I think it has to do either with the volume regulation of these cells, or it's also maybe metabolites in that very close structure, also in the free floating aggregates. I would think that you have, of course, osmolytes maybe that create like a hyperosmotic environment. I mean, that would be now a bit the hypothesis. And this is also why we now do also experiments where we from outside try to change the osmolarity of the medium in order to see whether we can basically maybe cancel out these effects or make them even stronger. Yeah.
Thank you.
Thank you. I think Joelle is next.
Hi, nice talk. So I guess I'm going to repeat what I wrote. Basically, I'm wondering how much of the history the cell or the nucleus keeps over time, and how long would that take to kind of resolve that? And the question is like, how dynamic are all that lamine structure in your system, and how long it would take for this, if you build something that's small and then it swells suddenly, maybe you have a lot of mechanical stress and energy there. And I wonder how long it would take for that system to just adjust to this new state, if you have any idea.
Yeah, that's a good question. I mean, we took out these cells, and then our approach was to image them relatively fast, of course. So that was because we didn't want to let them go much further in cell cycle, right? We thought, okay, we don't want to have growth of these cells, but we really want to look at a short timescale recovery, which was still half an hour, 40 minutes. I think much faster we can't really do that, unfortunately, because it takes a while because we have to digest the matrix. I mean, if we would start with the free-floating aggregates, of course, that could be shortened. Yeah. And I agree. I mean, what we have not done really to look at mechanic, I mean, I think I did that quite a long time ago with AFM, where we basically, it was stiffness, sorry. Yeah, but we don't have a time course after. I mean, that would be very interesting to see whether we get these mechanical changes also, that we see how the cells would recover from them. Because I would also think that once these cells swell, that probably the cortex probably adapts, and that is maybe also some part of it what we pick up with AFM when we now measure them. Yeah. But this, I think that would be interesting to do more time steps after to follow up. But at least we see that relatively short, we get a recovery, but after that we haven't really looked. And also, I see in your question in the in the chat that lamins, yeah, we did some lamin stainings, I believe, but not after taking them out. Yeah.
Okay. Thank you.
Okay. Well, I would like to thank Neil and Anna for kind of really great biophysical talks, and thank you everyone for coming, and we will see you in 2 weeks. All right, everyone, take care.
Thanks a lot.
Bye.